Sugar beet is a agriculturally very important crop plant. In 2021, there was 270 million metric tons harvested of sugar beet. It is a diploid species with 9 chromosomes, and it can readily crossbreed with all its wild relatives. This also makes the wild relatives of sugar beet the primary gene pool for crop improvement. The main aim of the study was therefore to understand the phylogeny of sugar beets and also the phylogeny of sugar beets and its wild relatives. I will talk a little bit about the data. So we sequenced a lot of beets. So in total, we sequenced 645 beets. We used whole genome sequencing data using Illumina short reads, and the coverages have been between 4 and 10-fold. The plant material we got from public seed banks, the IPK and the USDA, but we also got state-of-the-art breeding material from breeding companies and pre-breeding material from the USDA in Michigan. We sequenced 3 species. We sequenced Beta vulgaris, Beta macrocarpa, and Beta patula. And from Beta vulgaris, we sequenced the subspecies vulgaris, which is the sugar beet, then the wild ancestor of the sugar beet, the sea beet, and then we sequenced Beta danensis, Beta macrocarpa, and Beta patula. So for the sea beets, this is a representation of the sampling locations of all the beets. So you can basically see that we sequenced beets from all over European coast. So we sequenced the Mediterranean coast, and here the Strait of Gibraltar and all of the Atlantic coast. So we also have this as an interactive interface. So if you scan this, you get to our website and you can hover over all of the plant points, and you get basically more information about all the about the plants that we sequenced. The workflow that we have used is basically we did the Illumina sequencing as a first step. Quality control, we generated k-mers for all of the sequenced accessions using a tool by Ondorf et al. Called MESH. We then calculated pairwise Jacquard indexes, and with the pairwise Jacquard indexes, we calculated phylogenetic trees from the pairwise distances of all the accessions. And I will now go to the results. So the first thing you can see here in red are the branches that are sugar beet accessions. In green or yellow, you can see the Mediterranean sea beet accessions, and in blue, you can see the Atlantic sea beet accessions. And the branches here represent basically the original species annotation in the public seed banks. The first thing we realized is that we can separate the sugar beets from the wild beets quite clearly, but we also can separate Mediterranean from Atlantic sea beets, which also means that there has to be some kind of genetic barrier at the Strait of Gibraltar, because also when you look closer into the samples and they propagate over the sea, you can see that they are genetically quite far apart. And so there has to be some limitation of gene flow at the Strait of Gibraltar between Mediterranean and Atlantic sea beets. The second thing you can see here is, if I find this here, there are, for example, red dots here. So they have been annotated as sugar beets in the public seed banks, but when you cluster them, you see clearly that they cluster like in the middle of sea beet accessions, which should not be possible. And so we think that these are misclassified accessions in the public seed banks, which obviously has a huge impact, because if you order these seeds, you grow them, you just take one leaf and you sequence them, you basically have a different species you work with than you think you're working with, which has a huge impact on your downstream analysis. So basically we corrected all of these annotations in the public seed banks. And also here are some gray dots. These gray dots mean that they don't have species annotations in public seed banks. We also added these species annotations. The last thing I would like to talk about here is that when we look at the genetic distance, we always see that plants from far eastern Mediterranean coasts are the closest to sugar beets, which is why we think that the origin of domestication is in Greece or in the eastern islands of Greece. The only caveat of this is I showed in the beginning a map. We haven't sequenced many accessions that are not from Europe. So in the next step, we are at the moment sequencing accessions from Israel and other non-European countries to narrow this further down and get a better explanation about the origin of sugar beets that we have. We also included the other wild beet species in our analysis. And when you look at this here, you see here some sugar beets. Here the C beets, here are beta macrocarpa, and here beta adenensis. The nice thing is that we basically could separate all of the groups very well, but I have to tell a little story, because here you see a lot of blue dots in the adenensis accessions. And when I started working with this data, I always found C beets that have been genetically super far away from the other C beets. And it was like super stressful, because I was like, am I doing something wrong? Did we do something wrong with the sequencing? What is wrong? And only when we added then the adenensis, like the different subspecies, we saw that they cluster together perfectly. And like all of these accessions are most likely misclassified in public seed banks, which is quite problematic, but also the reason for this is quite clear, because when you go into the field and you look at these two plants, they look quite similar. So it's easy to mix this up when you collect plant material. But for us researchers, this obviously is crucial, because you don't know which species you get when you order seed material from seed banks, which is not very well. I also circled it, but that's too late now. We also did phylogenies of the sugar beet accessions, and I will circle in a little bit. In the beginning, I told you that we sequenced 30 accessions from plant breeding material, plant breeding companies. These companies are KWS, Strube, and Syngenta, and they represent state-of-the-art breeding material, like real breeding material that is used nowadays for sugar beet breeding. And the interesting thing is that in the phylogeny, they cluster directly together, like all of the plant breeding accessions, independent of from which company they come, from one clade in the phylogenetic tree, which also shows that we can basically resolve even fine phylogenies with this KMA-based method. And not only do the company accessions cluster together, but also if you look closer, you see that the accessions from the different companies cluster together. So we can cluster the Strube accessions, the Syngenta accessions, and the KWS accessions. So this is quite nice. And also here you see the pre-breeding accessions from the USDA in Michigan. The last thing I would like to show is we also included publicly available disease resistance annotations in our trees. So we extracted subtrees from our large trees that we have basically made and added annotations for the disease resistance. And this allows us, for example, if we want to have more information or sequence deeper some accessions that are resistant and susceptible to a disease, but are genetically close or close together in a phylogenetic tree, we can select this based on this data that we provide. And we provide this for, I think, 7 different diseases that we have publicly available disease resistances. So if you're interested in linking the genetic information and the disease annotations, this is a very good thing. So to get to the conclusion, KMA-based methods quite accurately resolve even detailed phylogenies. We have a genetic barrier between Mediterranean and Atlantic sea beets. We also see that many accessions are misclassified in public databases, and we linked phylogenetic trees with disease resistance annotations. Since then, we sequenced 700 additional cultivated and wild beet accessions. So we now have a dataset of 1,300 fully sequenced whole genome sequence beet accessions. We do this to further refine the phylogeny of beets, of white and sugar beets. And we also use this, for example, to identify domestication genes and domestication processes in beets, which is a paper that we release shortly. This is the publication. I'm very happy if you read it, but you don't need to scan it now. I have it on the next slide as well. Some acknowledgements. I would like to thank my supervisors, Juliane Dom and Heinz Himmelbauer, Nancy for library preparation of 1,300 accessions and also the sequencing, which is quite nice. My doc school for allowing me to be here, and Britta Schulz from KWS and Mitch McGrath from the USA for plant material. And yeah, thank you everyone, and I'm happy to answer all questions.
Thank you. One short question. Any questions? No questions. Okay.
So the first phylogeny you showed, and I know nothing about beets, this looked very unbalanced, or did I get this wrong? So
what do you mean?
I mean, like you have, I think the one before, yeah, like you have like a long branch, and then everything else is shorter. Like it sorts really nicely. I think you can compute like a statistic called a Coles statistic, and this would have pretty extreme coalesce value.
This you mean?
No, the one that you just showed. Yeah, this one.
Yeah.
Did you know if there's some like a demographic process, or why is it? Why does it look like this?
This is just the representation. Like the leaves are all that long. It's just like easier to see like this. So if you calculate phylogenetic trees, you can choose to either show like the direct distances in the cladogram or not, right? And for these figures, it just decided to show like this, but it does not look in reality like this. So there are different distances between the plants.
Okay, thank you.